Hello everyone. Today we are going to discuss a very important topic of pharmacokinetics, that is the absorption of the drug. Absorption of the drug is a process by which a therapeutic agent gains access to bloodstream and is ultimately carried to the site of action. It is usually associated with oral drugs and their absorption through the GIT. It also occurs by subcutaneous route, intramuscular route, and transdermal routes of drug administration. The drugs have to cross the biological membrane to get absorbed into the blood circulation. The biological membrane consists of a lipid bilayer separating different compartments of the body, with protein molecules acting as enzymes, channels, or carrier protein with which a drug molecule or a drug can crosses the cell membrane. There are different mechanisms of drug absorption, or you can say different processes of drug absorption, like passive diffusion, facilitated diffusion, active transport or active diffusion, endocytosis or exocytosis. Now we will discuss all these processes one by one. We are going to start with the passive diffusion. In this form of absorption, the drugs moves from the area of high concentration to the area of low concentration. Most of the drugs are absorbed by this passive diffusion, which is the movement of the drug from area of high concentration to the area of low concentration. This process does not involve a carrier protein. It does not require any energy. Many of the drugs get absorbed through passive diffusion. Most of the lipid soluble drugs readily crosses the cell membrane, and water soluble drugs get absorbed across the cell membrane through the aqueous channels. After passive diffusion, we have facilitated diffusion, and as its name suggests, in facilitated diffusion or transport, the drugs absorb through cell membrane with specialized transmembrane carrier proteins that facilitate the passage of large molecules of the drug. It involves the drug moving down the concentration gradient by the help of transport protein. No energy expenditure is required. This type of transport is also specific and saturable. The main objective of facilitated diffusion is that lipid insoluble drugs become lipid soluble by combining with the carriers. For example, iron binds with apoferritin and certain catecholamines enter the nerve cells by this facilitated diffusion process. There are many drugs that get absorbed inside the body with the help of facilitated diffusion, like corticosteroids, ethacryonic acid, and captopril. Here in this picture you can see that this is the carrier protein, and with the help of this carrier protein, drug get absorbed or crosses the cell membrane. Then we have active transport or active diffusion. It is defined as the process that involves the movement of molecule from a region of lower concentration to a region of higher concentration against the gradient or an obstacle with the use of external energy. So in active transport, special carrier proteins are required. It is an energy dependent process, and that energy comes from the hydrolysis of ATP. In active transport, drugs moves from the area of low concentration to the area of high concentration. Now there are many drugs that get absorbed inside the body with the help of active transport or active diffusion. We have a antibiotic that is known as penicillin. Then we have some anti-cancer drugs that get absorbed with the help of active diffusion, and the best example for that is 5-fluorouracil. And then we have digitalis glycosides, which is the drug used for the treatment of chronic congestive cardiac failure. In this picture you can see the two processes of absorption, passive transport and active transport. In passive transport, you can see that drugs moves from the area of high concentration to the area of low concentration. There is no requirement of energy. And in active transport, you can see that drugs moves from the area of low concentration to the area of high concentration, and this process requires energy. After that, we have another process of absorption, that is endocytosis. It is a process of capturing a drug from outside the cell by engulfing it with the cell membrane. There are two main processes of endocytosis. One is phagocytosis, that is also known as cell eating. This type of transport is utilized by large molecular weight drugs. And then we have penocytosis. Penocytosis is also called as cell drinking. This requires expenditure of energy. Fat soluble vitamins, protein molecules and folic acid enter the cells by the process of penocytosis. Then after endocytosis, we have exocytosis, and it is the expulsion of drug outside the cell. Many neurotransmitters, they get released inside the body from the nerve endings through the process of exocytosis. So that was all about the absorption of the drug. I hope the absorption of the drug and different processes of absorption are more clear to you. In the next lecture, we will discuss different factors which affect the rate of absorption. So stay tuned, subscribe to the channel and stay curious. Thank you.